The last topic in Chapter 11 in this lecture is actually not in your text, and it's a topic that I have found that students aren't really aware of at all, and it has to do with some basic function of the cell. And the scientist who discovered this won the Nobel Prize in 1999, which is where I got the slides and the figures for this. For the most part. It's called signal hypothesis, although now it's not merely a hypothesis, it's something that's well supported. But in 1999, Günter Blobel won the Nobel Prize in physiology or medicine for the discovery that proteins have intrinsic signals that govern their transport and localization in the cell. So you've heard before that, for example, proteins that need to be secreted from the cell are delivered into the rough ER, and then they travel by vesicle to the Golgi, and then vesicles take them to the cell, to the plasma membrane, where they are secreted. But how do some proteins know where they're supposed to go, and some just stay inside the cell? And so this work that Günter Blobel started and has been added onto since then explains that proteins have built into their amino acid sequence a signal that governs how they're going to be moved around in the cell, and then if they're going to be secreted, how that governs the secretion from the cell. So his work was initially started in the 70s through the 80s, but he didn't win the Nobel Prize till 1999. Which is pretty typical. There's typically a lag time between when a discovery is first made and when the Nobel Prize is awarded. The statement here is something that we're going to spend some time looking at. It says, although all translation begins on free ribosomes, comma, I just want to start with that sentence. You may be aware that there's two types of ribosomes. One that's called bound ribosomes. Those are attached to the rough ER surface. That's why it's called the rough ER, because it's covered with these ribosomes that make it look bumpy. And then there are free ribosomes, which are in the cytosol. But it turns out that all ribosomes are exactly the same. And the ribosomes that are on the rough ER aren't always on the rough ER. It's not a permanent attachment of a ribosome to the rough ER. So ribosomes are constantly moving from being in the free state of being a ribosome to being bound. So free and bound ribosomes is just a matter of where a ribosome is right now as to whether or not it's free or bound, because a free ribosome can later be a bound ribosome, and then later after that it can become a free ribosome again. So ribosomes are dynamically moving from the cytosol to the rough ER and then back to the cytosol in a very dynamic way. So there aren't fixed ribosomes that are permanently attached to the rough ER, which is probably the way a lot of students think of it, because that's kind of the way it's presented initially, but it's kind of an unintended assumption that's made that the ribosomes permanently stay on the ER, for example. So what this statement is saying here on the slide is that all translation actually begins on free ribosomes. But once the ribosome starts to translate an mRNA, a little bit of the protein product will start to stick out of the ribosome. And so if that protein is destined or needs to be secreted, delivered to another organelle, or inserted into a membrane as a transmembrane protein, the very beginning of that protein will contain what's called a short peptide signal sequence. And it is a certain sequence of amino acids at the very beginning of the protein that's synthesized at the very beginning, and that will signal whether that protein needs to be delivered to a different location instead of just staying in the cytosol. So when this short signal sequence, which is typically about 15 to 30 amino acids and includes the start methionine, when that little section of the protein starts to stick out of the ribosome, there is a protein complex in the cytosol called the signal recognition particle, which will stick to that signal sequence and move the entire ribosome with its mRNA and with its emerging protein product to a membrane channel on the rough ER. And the protein synthesis will then continue at the rough ER. Now the ribosome is called a bound ribosome, and the protein will be threaded in through a pore in the rough ER membrane, and then when it's finished being synthesized, it's released from the ribosome and it's released into the rough ER lumen or space. And at that point the signal sequence is trimmed off. So you don't see the signal sequence in the final protein product typically because it's been trimmed off early in the process. This picture shows really a snapshot of events that corresponds really just to one ribosome doing its translation of one protein. But they've tried to put all the events on one picture, so it looks kind of weird. But if you start from left to right and you sort of number the ribosomes in sort of a time sequence, this is time 1 on the left, you see a free ribosome. That's what they're trying to show here because it's not attached to the ER. The free ribosome starts to translate the mRNA, and a little bit of the protein is being made here, shown as a little ziggy-zaggy. And as that sticks out of the ribosome, it will cause the signal recognition particle to attach to that and then move the entire ribosome to dock on the rough ER at a membrane channel, shown here in purple. And you see how the protein is now threaded through that membrane channel, and it continues to be synthesized. Now the ribosome stays still at this point because it's attached to that membrane. So the mRNA is basically moving instead of, I know you think of the ribosome moving along the mRNA, but actually it's probably the mRNA that's moving. But in any case, the ribosome is working generally in the direction of the 3' end of the mRNA, translating the codons. And as that continues, the protein continues to be threaded through, and you can see it folding as it enters the lumen here. And you can see also the signal sequence, the signal peptide has been trimmed off pretty early on. So by the time the protein is released here on the right side of the picture, the mature protein folds, the signal sequence has already been trimmed away. And then the ribosome, it's important that you see here, when the ribosome is done translating this protein, it releases from the rough ER membrane. So it becomes a free ribosome again. So ribosomes, first of all, always start off free. If they are translating a protein that has a signal sequence, then only then they will become bound to the rough ER. And when they're done translating that protein, they become free ribosomes again. So it's a very dynamic movement of ribosomes to and from the rough ER. Since Global's work in the 70s, they have figured out that there's a lot other signal sequences, not just the ones that direct things to the rough ER, but other signal sequences that can direct things to be delivered into the nucleus or into chloroplasts or into other things that aren't part of the endomembrane system, like the mitochondrion. But that came after. I mean, Global is the one who came up with the very first example of, a type of signal sequence. And from that work, more, extensive work has been done to find out that lots of proteins have different kinds of signal sequences that target them to all kinds of organelles and locations in the cell. But because the eukaryotic cell is all about compartmentalization, if you have a faulty signal sequence, meaning if you have a mutation that affects the amino acid sequence in the signal region, then that can cause a protein to fail to be delivered to the correct cellular location. So the protein itself might be completely functional, and remember that signal's been trimmed off, so sometimes you can't even see it in the protein product. But if it doesn't end up in the right location, it might not be able to perform its function correctly. So a faulty signal sequence causes a mislocalization, meaning a protein that can't be delivered to the right place in the cell, and then it can't do its job. So you have, for example, you could have a mutation where a protein that's supposed to be delivered into the lysosome, which buds off of the Golgi, but if it doesn't ever get into into the Golgi, if it never gets delivered to the rough ER at first, then it ends up, you have a digestive enzyme that's sitting in the cytosol, which actually could be very harmful. But you have some other examples of disorders that can be caused, among many mutations that can cause these things. One mutation that can cause these disorders would be a mutation in the signal sequence. So I gave you a couple examples here. Primary hyperoxaluria is caused by a deficiency of a peroxisome enzyme. And the one reason that you might have a shortage of that enzyme is that your cells are making the enzyme just fine, but the signal sequence is mutated so it's not being delivered to the peroxisome. It just ends up in the cytosol. So it can't do its job there. Enzymes have to be in the correct organelle to do their specific job. Familial hypercholesterolemia, that's always a hard one for me to say, and cystic fibrosis can be caused, among other types of mutations. One mutation that can cause these would be a mutation in the gene that affects the signal sequence, because both of these disorders are caused by problems with transmembrane proteins. So you have maybe a transmembrane protein that ends up in the cytosol instead of being inserted into a membrane. So you can get a hereditary disease that's associated with a faulty signal sequence. Even though the protein that's finally produced functions fine, it's not delivered to the correct organelle or cellular location. So that wraps up Chapter 11 lecture.